I'm going to just, I'm not going to talk too much about a scientific approach or a new therapy. I'm going to talk about the general problem of how we get new therapies into the clinic. What we've worked with extensively at Mayo Clinic is, since I've been there, is using measles virus as a cancer therapy. And you can see this photograph of a boy here. He turned up in a clinic some years ago with a very nasty retroorbital tumor. It turned out to be a lymphoma. And he wasn't given any treatment because he had measles at the time that the diagnosis was made, and the tumor went spontaneously away. And there are many case reports of patients who got a virus infection, virus got into the tumor, got rid of the tumor, and so that essentially is what we're trying to do. And of course, with measles, we don't want to use the wild-type virus because it causes measles, which is an unpleasant illness. But we're fortunate to have the vaccine strain that we all give to our kids, and we know it's very safe. And it turned out, serendipitously, we started looking at measles, that pretty much any human tumor cell that we took and cultured and exposed it to measles was immediately killed by the virus. And that's turned out to be the case with quite a few viruses. But the problem is, we have all these new agents coming through, and we have to take them through to clinical testing. And that's what I want to talk about. So, basically we have our preclinical research, and this is a completely new therapeutic paradigm. So up until recently, all new drugs were made by pharmaceutical companies. Okay, they had robots and huge high-throughput assays and libraries of hundreds of thousands of compounds, and they would find a compound that actually did the job. But then came this sort of gene therapy thing. With gene therapy, we know which gene we want to put into which tissue in the body, and we know what we expect the outcome's going to be. So there's this armchair phase of drug development now, where provided you know sufficient, you can design and build, and then you've got a therapeutic that you can test in animal models. So you keep going around this preclinical cycle until you've got something that really works, at least in the animal models, be they mice or rats or whatever. But then you obviously want to test that in the clinic. That's where things really can make a difference, and you go through these different phases of clinical testing, and you keep going around that loop. But what people often forget or don't know about is that to get from the lab to the clinic is a huge effort, and it's a huge journey. You cannot just take something you made in the lab and give it to people and keep your fingers crossed. There's a whole regulatory process. Al works for FDA, and they deal with this the whole time. But, as far as they're concerned, if you're going to put a virus into a patient, you better have a manufacturing facility that is a pristine facility run by very good people who can ensure that every product that comes out of there is pure and clean and the correct identity and safe to give to patients. You have to perform toxicology testing and pharmacology testing, and you have to do it in whatever species FDA say. If you go to FDA and say, well, we'd like to do this tox testing in mice, and we think that'll be fine, and they say, no, you have to do monkeys, then you have to do monkeys. I mean, when the FDA say jump, you say how high. You don't actually argue with them. And if you do argue with them, they usually tell you, okay, you can do whatever you're suggesting in addition to what we told you to do already. So you learn very quickly that it's not productive to argue with FDA. And then, of course, you have to develop your clinical protocol, and that drives everything. If you've developed a virus and shown in animal models that it's good for cancer of the breast or cancer of the head and neck, then your clinical protocol has to specifically address that indication. And when the FDA tell you to do the toxicology test, they're going to consider that protocol. And if you're injecting it into a breast tumor, they're going to want you to do injections into the breasts of whatever your experimental animal is going to be to know that it's safe when administered in that way. And you have all these regulatory hurdles in addition to FDA, the Food and Drug Administration. There's a public forum in the States for all new gene therapy, because many years ago, when gene therapy was first sort of coming on stream as a clinical intervention, it was clear there was a lot of anxiety. I mean, people were worried about creating monsters, going against nature, changing the genetic makeup of people for future generations. And so there was a public forum established in the States. So every new gene therapy trial has to be publicly discussed. It's reviewed by peers. You send in the proposal, gets reviewed by your colleagues, and then you have to go to this public meeting and you have to defend it. And anyone from the public can come and witness that and join in the discussion. And then in addition to that, you have the institutional boards, the ethical review board at the institution where you work, and the biosafety committee. So you have to make sure you get all these regulatory approvals. And it takes a lot of paperwork. If you don't have an appetite for this paperwork, then you just can't do it. That was the paperwork we submitted to FDA for the first virus we took into the clinic, just to open discussions with them. That wasn't for the approval. That was so we could have our initial discussions. And the amazing thing is they read it all. And, they knew it inside out when they had the discussion with us. So this whole process is, it's just don't forget it. It's very, very important. And it takes a lot of money. It takes a lot of time. And it takes a team approach. I mean, no one can do this on your own. If you're the scientist who created the product, you've then got to interact with the clinicians, the manufacturing people, the tox farm people. You've got to get through those regulatory hurdles. And it really does require a team approach. And for the manufacturing, I'll just give you a sense of that. This is a facility, it's a floor plan of a facility that we have at Mayo Clinic in academia. And, in the past, just to continue with what I was saying about the new paradigm of design, build, and test. So now that you don't actually need a pharmaceutical company to be involved in the production of your new product, many academic centers have reached the point where they've got a lot of really exciting new things that they developed in the lab, but they don't have the infrastructure to be able to take it and test it in the clinic. They don't have the manufacturing facility or the tox capability. And so, this becomes very important. And this is the sort of facility you need, where you're controlling all the airflows, what the particle counts are. You've got a very controlled environment because that's required from a regulatory perspective. The people working in there have to wear bunny suits. They're the dirtiest thing that ever goes into the facility. And so you have to have a really tightly controlled environment. You have to do all this QC testing to make sure that it's safe to administer to people after it's been made. And then for the tox testing, that takes another team with a very specific approach. Tox testing is really quite different than regular science. If we ever take a tox study to the FDA and we say, we did this, that, and the other to these animals, we measured the hemoglobin, and it was 10 on the first day, it was 14 on the second day, they say, hold on a minute, prove to us that your machine was actually working at the time that you measured that hemoglobin. Show us the records on the maintenance of that machine for the last 2 years. Show us the records that prove that the people who are running the machine actually knew what they were doing and knew how to run it. You have to document all this stuff, which, in a regular lab, you don't do. So it's a very different approach. It's called GLP, Good Laboratory Practices. And it requires a kind of crack team of people who are familiar with that. So that really is my message to you, is that, translation is a big deal. Don't be so impatient when you read about something exciting in the newspaper and it hasn't turned into a cure for cancer tomorrow. It takes a lot of time to get through this whole process. And I'll leave it there. So, thank you. Len asked me to just tell you about this, so I will. I Am Legend. I don't know if anyone saw that movie, but it was a movie that came out a couple of years ago. Scene one was 2009 TV interview with a cancer researcher, Emma Thompson, who had engineered a measles virus, of all things. And it was being used to treat cancer. And she said in the TV interview she gave at the beginning of the movie, she says, we've treated 10,009 patients, and they're all cured. And I tell you, that scene I just love. I just wanted to be Emma Thompson, as soon as I saw it. But then they fast forward to scene 2, and it's 3 years later in New York City. And everyone's dead. except for Will Smith, who's the central character. And it turns out that what's happened is that measles virus therapy mutated and it spread through the population and killed pretty much everybody, except that later we discover there are some survivors. They come out of the New York subway at night and they're trying to get Will Smith. And this is what they look like. And so Len said when I showed him this photograph, he said, "Is that the first patient you treated with measles at Mayo Clinic?" So anyway, this was a problem for us, and I just want to update you that our trials are moving along slowly but steadily, despite the availability of that movie on DVD. And we've got no cures yet, but we're seeing activity in ovarian cancer, and we've seen no dose-limiting toxicities and no zombies. And there I will really end. So